Scientists have developed a new antibacterial compound that can specifically target Mycobacterium tuberculosis, or MTB for short, the bacterium responsible for tuberculosis infection. This compound, called CMX410, overcomes prevalent specificity and bacterial resistance issues that have been constantly faced over the history of tuberculosis treatment. When CMX410 is released, it uses a specially engineered electrophilic domain to target one of the enzymes in MTB called PKS13, which is responsible for cell wall biosynthesis. A covalent bond irreversibly inhibits the enzyme, leading to disruption in cell proliferation and effectively resulting in widespread cell death. So that sounds great, but what is tuberculosis and how does it work? In terms of global impact, there is an estimated 2 billion people infected with TB, either actively or asymptomatically. This includes around 1 to 2 million people who are infected with the active form. On top of this, there are approximately 10 million new cases every year, with 1.5 million of them resulting in death. This is currently the leading cause of death by a single infectious agent, higher than HIV and malaria. MTB enters the body through inhalation and travels down into the lungs. Here they are recognized by alveoli macrophages, which immediately act to surround and engulf the pathogens. This is where MTB hijacks the system. Under normal conditions, in order for phagosomes to degrade pathogens, lysosomes must bind to these macrophages, releasing various proteins and ions that function by dropping pH and activating hydrolytic enzymes and reactive oxygen and nitrogen species. This is usually enough to kill any invading pathogen. Unfortunately, the response for tuberculosis is different. MTB is able to evade the immune system by blocking this phagosome-lysosome binding using virulence factors such as protein kinase G and SAP-M, which both work to inhibit host proteins and lipids and disable phagolysosome binding. MTB then adapts and survives inside the macrophage intracellular environment by 1. Upregulating stress response genes, which enable survival under hypoxia, low pH, and other host-imposed stresses. 2. Utilizing host lipids for energy via its own modified TCA cycle. And 3. Reducing metabolism and entering a non-replicating state, enabling it to persist in an inactive form in the body until an optimal time to infect. This occurs when the immune system is compromised, for example by AIDS or aging. MTB releases itself from inside the macrophages and spreads across the lungs. Here, it induces a strong immune response, which can lead to scarring and pneumonia in the lungs, and spread via the vascular system to almost every other organ in the body. This is where the infection progresses to systemic miliary TB, reaching the kidneys and causing sterile pyuria, meningitis in the brain, Addison's disease from the adrenal glands, and hepatitis in the liver, just to name a few. So what makes this bacteria so difficult to treat?
MTB is hard to kill for three main reasons. First, it can evade the immune system by hiding and living in our own immune cells, especially macrophages, as explained earlier. Secondly, MTB grows relatively slowly and can enter metabolically quiescent states. Most conventional antibodies often target cell proliferation with either targeting cell wall synthesis or transcription translation, so a slow-growing, intermediately active pathogen is inherently harder to sterilize. Thirdly, MTB has a very unusual waxy cell wall, with the outer layers being tightly packed with mycolic acids, which are extremely long fatty acids, around 60 to 90 carbons long. These mycolic acids are extremely hydrophobic and have a very low permeability. This causes commonly used hydrophilic antibiotics to not go through very easily. Anchored on top of these mycolic acids are glycolipids, which further decrease permeability and are able to modulate immune recognition. This waxy cell wall slows the entry of many antibiotics and helps the bacteria resist stresses like reactive oxygen and nitrogen species. Put together, you have a bug that hides in cells' walls, wears chemical armor, and refuses to grow fast enough for our drugs to hit it efficiently. The mycolic acid synthesis pathway begins in the Fas1 enzyme, where it converts acetyl-CoA and malonyl-CoA into a medium and long acyl-CoA. These then split up, where one enters the Fas2 cycle, where it transforms into a very long myrmecollate, acyl ACP. The second pool, with carbons around 24 to 26, is carboxylated by the enzyme ACCD4/5 to form a 2-carbyl acid CoA, which will become the alpha branch of the mycolic acid. Then the FAD-D32 enzyme activates the myrmecollate, forming a myrmecollate AMP, and then loading into one arm of the PKS13 enzyme. The 2-carboxyl CoA form ACCD4 is loaded onto the second arm via the PKS13 acyl transfer domains, AT. Within the red box, PKS13 domain performs a Claisen condensation, joining the 2 acyl chains and releasing CO2 to form the mycolic acid backbone. So, CMX410 inhibits PKS13 by chemically sabotaging its catalytic machinery. PKS13's ability to create the mycolic backbone reaction depends critically on the acetyltransfer or AT domain and its catalytic serine, which loads the alpha branch onto the appropriate ACP arm and positions the substrates for condensation in the ketosynthase site. CMX410 contains an aryl fluorosulfate or a SuFEx electrophile. When the inhibitor binds inside the PKS13 AT active site, the catalytic serine 801 performs a nucleophilic attack on the sulfur at the center of the fluorosulfate. This displaces the fluoride and forms a covalent sulfate ester between the CMX410 and the serine 801. This permanently distorts the AT active site. The result is irreversible mechanism-based inactivation, with PKS13 no longer able to load acyl substrates or participate in condensation. Without this step, there is no mycolic acid intermediate forms and shutting down the entire mycolic acid biosynthetic pathway, and ultimately collapsing the mycobacterial outer membrane and causing widespread cell death.
So how much of a breakthrough really is this new compound? The first MTB treatment was established in the 1980s, which involved a 6-month course of a variety of drugs. This was initially very effective, however, MTB quickly acquired resistance. In 2012, a new drug called betaquilene was approved. This drug selectively inhibits ATP synthase and is effective against active and resting MTB. Unfortunately, however, this drug has been found to cause liver toxicity and central nervous system toxicity. Bacterial resistance has also been observed. Another contender, delamanid, was approved in 2014. This drug suppresses mycolic acid synthesis, resulting in higher specificity. However, similar side effects to betaquilene have been observed. Finally, presemanid is an even newer drug, approved in 2019, and works by producing reactive nitrogen species to disrupt bacterial DNA and proteins. This, however, is limited by serious adverse cardiac effects, which have recently been displayed. This new SuFEx-based compound, on the other hand, has shown extremely positive results. Although they are still preclinical, mouse models have shown a greater than 100 times reduction in bacterial resistance compared to other approved cell wall inhibitors. They have also displayed no adverse effects in rat toxicity studies using maximal dosages of 1,000 milligrams per kilogram per day. Overall, the irreversible nature, paired with the MTB-specific PKS13 target, for which there are no existing mammalian homologs, provides for strong pharmaceutical prospects in antitubercular treatment.